Welcome to week 2 of anatomy. Today's topic is the cell. And by looking at the image here in front of us, we can tell that the cell has a lot of different pieces and parts. And each of those pieces and parts has a specialized function that it carries out. And you'll read about each of those as you read through your reading assignment. What I want to spend a couple of minutes talking about here is, though, the cell membrane, or the plasma membrane. And it is a very complicated structure. If we look at that a little bit closer, we would see that it's what we call a phospholipid bilayer. The bilayer part means that there are two layers of phospholipids. And these sort of look like a sperm with two tails. Heads to the outside, tails to the inside. And the function of the plasma membrane, or the cell membrane, is to determine what comes into the cell and what goes out of the cell. So to enter or exit the cell, you have to be able to get past this membrane. Looking here at it, it has a number of proteins, all these purple structures, and they have a number of different functions. So let's look at some of those functions of those proteins. The first one here is transport. So if you are a molecule that is fat-based, so you're made out of fat, then you can move through the phospholipid bilayer very easily, just basically wiggling through the fat. And that's not a big deal because fat dissolves fat. If you are not a fat-based substance, to get across the membrane it's a little more tricky. So the transport proteins can actually pump things into or out of the cell across the cell membrane. Sometimes that takes energy, sometimes it doesn't, but either way, they are basically a door in the membrane that allows non-fat-based molecules to pass through. We won't spend a lot of time talking about functions B and C there. It's fairly self-explanatory. They can be a receptor for a message. They can be an attachment protein to something else around in the environment. And then D, they can function as an enzyme. So those are basic functions, but not overly interesting from my perspective. Function E, intercellular joining, is a little bit more interesting in that this is how cells attach to each other. And in your reading assignment, you'll get to those different attachment types and understand what we're talking about there. It'll be things like gap junctions and tight junctions and desmosomes, things like that. Different ways cells attach to each other. What I really like to focus on here is function F, cell-to-cell recognition. And so if we go back and look at that membrane, what we see are lots and lots of glycoproteins. And the glycoproteins actually have some carbohydrate chains attached to them. That's all of those purple, or rather green structures sticking up out of the membrane. And those are all carrying out this function of cell recognition. So this is really important because the immune system protects our bodies by reading name tags. And as long as it reads the name tag and recognizes it, it leaves it alone. So if it comes along and finds a red blood cell, it says, red blood cell. I recognize you, no problems. Bone cell. Yes, I recognize you. Liver cell, I recognize you. Muscle cell, I recognize you. You get the picture. If it comes along and reads, let's say, a bacterial cell, it says, I don't know what that name tag means. I don't recognize it. And so the response of the immune system is then to attack it and kill it. So basically, if it recognizes the name tag, it leaves it alone. If it fails to recognize the name tag, it tries to eat it. For the most part, this works out very well. But those of you who have allergies would understand exactly what we're talking about when we say that sometimes the immune system gets carried away with itself and makes mistakes. For example, if you're exposed to a particular allergen, let's say dust, mites, maybe cat dander, maybe pollen this time of year might be becoming an issue. Whatever you might be allergic to, some of you perhaps latex or penicillin or any number of other things. Basically, those things would be foreign particles in the body. And when they enter the respiratory system, the immune system would recognize them as foreign particles and start to attack them. And up to that point, that's perfectly fine. The problem becomes when the immune system overreacts and fails to recognize your respiratory system as belonging to you, and starts not only attacking the original trigger, allergen, but also attacking your respiratory system at the same time. This leads to sneezing, coughing, shortness of breath, the watery eyes, the runny nose, all those normal allergic reactions. Those are all because the immune system is simply getting carried away with itself and failing to recognize cells that belong to you and properly leave them alone. The treatment for an allergy response is typically some sort of antihistamine kind of product which turns off the inflammatory response. So really you're turning off the immune system's ability to recognize me and not me so that you don't suffer that problem. But you are then relatively unprotected from things that truly don't belong. So treatment of allergies can be a double-edged sword in that it can alleviate your symptoms, but it may also leave you more susceptible to real problems. So that you have to find a balance there somewhere with that. Some of the functions that go on within a cell or perhaps across the membrane of a cell, one of those is called diffusion. Diffusion is basically the movement of particles from an area of high concentration to an area of low concentration, because really all chemical and biological systems want equilibrium. They want to be balanced. They want to be equal everywhere. So our example here is a dye pellet. A little purple crystal of something, let's just say potassium permanganate for the fun of it. That's a purple crystal that will turn water purple like this example. And when we first put the pellet into the beaker of water, in the pellet, the color of purple, the molecules, are in high concentration. In the beaker of water, they're in low concentration. When we first put it in, there aren't any, so that's as low as you can get. Then, because those water molecules are moving around always in that beaker, or always in our body, they will bump into things. And if it can be torn apart, as you'll see here, you see diffusion occurring in the middle beaker. And basically, as the water moves around, it dissolves the pellet, basically breaking the chemical bonds between the structures there. And then as the water moves, it carries, in this case, the visible color with it. So at the end of this period of time, we no longer have a pellet with high concentration. We have all of the beaker's water equally concentrated with the purple color. So diffusion again, moving from high concentration to low concentration, seeking equilibrium. If we were in a real classroom setting, we in the classroom as humans would be in relatively high concentration. And as soon as I told you we were done for the day, you would immediately start diffusing out of the classroom. And it can be a very quick process. Within a couple of seconds after the announcement that class was over, there would be roughly the same concentration of students inside and outside the classroom. Diffusion can actually go across the cell membrane. So as I mentioned earlier, if you are a fat-based substance, you can very easily diffuse across the membrane because, again, fat dissolves its way through fat, no problem. Another option might be those proteins that function as doorways. And here we can get into carrier-mediated facilitated diffusion, basically meaning it takes a protein opening and closing a door to move things back and forth. So the protein would be the carrier. Channel-mediated facilitated diffusion would basically be just open a door, leave it open, and if you're small enough to go through, you'll go through. If you're too big, you can't. And our bodies have both of those types of proteins there as well. Option D here is called osmosis. And osmosis occurs with the diffusion of water. So strictly speaking, osmosis is the diffusion of water across a membrane, in this case across your cell membrane. You can think of your skin of your body as a cell membrane for your entire body. It works the same way. The particular protein there that allows for the diffusion of water is called an aquaporin. And you would recognize the effects of this if you ever stay in a swimming pool for a couple of hours Or if you swim in the ocean for a couple of hours, what you'll find is that you'll find movement of water either into the body or out of the body through the aquaporons in the skin. So osmosis is something that is moving water. And the reason why you would move water is, think of osmosis basically as channel-mediated facilitated diffusion for water. So in example C, we could move some sort of, let's say, sugar or other substance through the membrane if it was a big enough opening. But let's say what we were wanting to move across, let's say it was salt, for example. So we had a very high salt concentration outside the cell, a very low salt concentration inside. Diffusion then says, let's move into the cell. And because salt is a non-fat-based substance, it can't just diffuse its way through the membrane. It has to go through a protein channel. And in this case, salt would be too big to go through that channel. So if the product that is in higher concentration is too big to go across, water typically will go across in the opposite direction to balance it out. So if you were in the ocean, let's say, what would be saltier, you or the ocean? The correct answer would be the ocean. So the question now is, because I've already told you salt cannot go through these membranes, which direction is water going to go as part of osmosis? Is it going to go from the body to the ocean or from the ocean to your body? A simple rule here is that water always follows salt. Wherever salt goes, water goes too. Wherever salt is, water travels to it. So because the ocean is saltier than you are, water will go through these aquaporons from your body to the ocean. And your body is then trying to balance out the ocean. Now clearly you can't do that because there's not enough of you to balance out the ocean. And this is a common cause of death for being adrift or lost at sea, is dehydration due to the salt pulling water out of the body relatively quickly. If you spend very much time in a freshwater swimming pool or a bathtub, you would also notice that a pruning or shriveling appearance of the skin takes place, just like it would be if you were in the ocean for an hour or two. But in this case, the fresh water is less salty than you are, so that means that instead of water going from you to the outside environment, water is actually coming from the outside environment into you. So this means instead of losing water and dehydrating in the ocean, in a freshwater body of water, you're actually swelling. And it appears the same kind of pruning of the skin in both scenarios. In the case of dehydration in the ocean, you're simply losing water in some parts of the skin faster than others. In swelling in a swimming pool or bathtub, what you're doing is you're taking in water unequally throughout the skin, some parts losing it faster than others, and it gives that some areas collapsed, other areas looking a little bit ridged. Either way, your body's trying to balance out the water content. Now clearly you can't balance out a swimming pool either, so this can lead to a problem. You can actually have someone that can die of drowning without their head ever going under the water. So perhaps you're asking yourself, how is that possible? And have you ever heard of anyone drowning without their head going under the water? And the answer is probably not. The reason for that is your kidneys will take that extra water that comes into the body and will form urine from it and say, this is too much water, get rid of it. So you typically don't spend the, let's say, 5 to 6 hours that it might take to drown in a freshwater pool without going to the bathroom and excreting that urine being produced by the kidneys. So as long as your kidneys are working properly, that wouldn't be a problem. I would recommend, though, that a diabetic, who by definition has faulty kidneys, not spend long periods of time in freshwater pools or baths, because you can have a buildup of water which could back up into the lungs and cause problems. If a diabetic wants to spend time in a swimming pool, they should probably do so in a saltwater pool, which would not have that movement of water into the body. So let's look at this concept here in a different example. Let's say we have this U-tube of water, and not the same kind of U-tube you're watching this video on right now. And we've got that membrane in the middle. And if the membrane opening is big enough, the sugar molecules in this case will go from the right side of the tube in high concentration to the low side of the tube, the left side, until both sides are the same. Would this be osmosis or would this be diffusion? We had sugar going from high concentration to low concentration, so that's diffusion, and it doesn't become osmosis because the sugar moved more so than did the water, because the pore in the membrane was big enough. If we look at this example, again we have a high concentration of sugar on the right side, low concentration on the left, but the pore size of the membrane is much smaller, so sugar, like we mentioned with salt, can't go through. That means our only option is osmosis, so diffusion of water. In this case, the water is in higher concentration on the left side, so it will diffuse across the membrane to the right side where it was in lower concentration until the concentration of water gets the same on both sides. And what you see here at the end is that the water actually moved against gravity. Gravity wanted both sides' volume to stay the same, but osmosis said, no, that's not an option, because we have to get our concentrations the same, and we'll move against gravity if we have to. If we look at human cells and what they look like in these differing environments of salt concentration, we have some names for that, and the first is an isotonic solution. So in an isotonic solution, and these are red blood cells by the way, the red blood cell looks like it's supposed to. Somewhat like a donut that you didn't quite get the donut hole punched all the way out of in the middle. And so an isotonic solution basically has the same salt concentration inside and outside the cell. So you don't really see a significant movement of water into the cell or out of the cell. It's balanced. And most animal cells want to be in an isotonic situation. That's the happy situation. Option B here, a hypertonic solution, occurs when the salt concentration outside the cell is greater than the concentration inside. And because salt can't come in, water has to osmose out. And so what you see is the cell shrivels and collapses as the water leaves the cell, trying to balance out the external environment. Now, if what we wanted was picture A, for that cell to slide through capillaries one cell at a time, how well is the hypertonic cell going to slide through? Or is it going to be more likely to clump? And clumping blood cells then results in loss of blood flow. So this can be a real problem, and it often in humans is demonstrated with the idea of a dehydration headache. Whether it's because of excessive alcohol consumption the day before, or you simply haven't consumed enough water in your experience, when those cells start to collapse, you start to have blood flow issues to the brain, and the brain becomes oxygen deprived. It then expresses itself perhaps as a headache, and a dehydration headache is certainly a nasty kind, and the only solution for that is to consume more water, to get that environment back to an isotonic solution, so that the inside and outside concentrations of salt become the same. So really the solution to a hangover, other than obviously don't consume the alcohol to start with, would be to rehydrate, and that would take care of the headache problem. Scenario C here is a hypotonic solution, and in this case the cell is saltier inside than is the external environment. So water starts moving into the cell, trying to balance it out. You see the cell starts to swell. So from a red blood cell perspective, there are two problems here. One is that now larger cell has a much harder time passing through the very small blood vessels. Problem B is that if it swells much more, it will actually explode or lyse. At that point, the cell has died. Let's say it's 100 degrees outside. You've been outside working hard, mowing the lawn, something like that, and you're really thirsty. And you decide you want to drink a quart of something, and you're going to do so within a 5 to, let's say, 7, maybe 10 minute time span. So a pretty significant amount of fluid intake in a short period of time. You have available to you a nice cold quart of water. You also have as an option a nice cold quart of Gatorade. Pick whatever flavor makes you happiest. Which one of those would you drink and why? Some of you are perhaps saying you would drink the water. Others of you are saying you would drink the Gatorade. The important question is, why did you choose that choice? Let me tell you what I would do. I would drink the Gatorade for a couple of reasons, but most importantly, because it is an isotonic solution. It is roughly the same salt concentration as your cells are already. And I keep saying salt concentration. Keep in mind it could be sugar concentration or anything else that's dissolved in the liquids of your body. Gatorade is roughly the same concentrated level of stuff as your cells. So when that fluid enters your stomach, it would present as an isotonic situation. So not a significant movement of water into or out of the cells of the stomach. You want it to be absorbed, obviously, but the stomach is not the place for that. Those of you who said you would drink the water, here's the reason why that's a bad idea. If you do that, you will discover that you feel very nauseous and uncomfortable afterwards because you have presented your body with a hypotonic scenario. The cells of your stomach are saltier than that plain water you put in. Then they start absorbing that water very rapidly and then start to swell. Basically, your stomach cells are swelling. That expresses as pain, and really maybe that pain, that nauseous feeling, is your body's way of saying, the stomach cells are drowning. Stop. And if you were to vomit out that solution, it would at least stop the cells from swelling anymore. And that's the whole reason why Gatorade was invented in the first place. And now we have lots of different rehydrating drinks out there, and all of them have the idea that you can rehydrate without messing up the tonicity of the stomach and the body. And so you're putting into the body roughly the same concentration of stuff that it already has, and then it stays as a balanced situation. So you can restore water volume. You can also restore any kind of salt that you've lost. In the case of Gatorade, it restores some sugar so you have more energy, tastes a lot better, and then you put some color into it so it looks like something that's worth paying that kind of money for. What we see here is a process called active transport. And I'm not so much concerned with the different steps and learning each particular step, but what we've been talking about so far with diffusion and osmosis are what we call passive transport. Passive means we're not using energy. It happens for free. We don't have to put effort into it. Active transport actually requires energy. So active transport actually becomes important when you have a scenario where you don't want to reach equilibrium. You want to have a higher concentration on one side and a lower concentration on the other. Let's say your cells want to have 5 parts per thousand sugar inside the cell. In the blood, let's say you had 3 parts per thousand sugar, so diffusion would only get us to 3 parts per thousand inside the cell. And in this case, that's not what we want. We want it to be higher. So to get it higher, beyond equilibrium, we have to invest energy. So what you see here in steps 1 through 6 is the investment of energy in the form of ATP, pumping something from one side of the cell membrane to the other, creating a higher concentration on one side, a lower concentration on the other. And that takes a pretty significant amount of energy. So a significant percentage of the total energy consumption of your body is actually utilized in active transport, moving things back and forth against equilibrium. So those all work nicely, but what if the particles you're trying to move back and forth are simply too big to go through the membrane, or too big to go through any available transport protein? Then we need another option. So this is an example of what we call endocytosis, cell swallowing, moving something into the cell from outside that was too big to go across any typical way. So what will happen, as you see up there in step 1, is the cell will form a little hollowed indention in the membrane and move that particle into that little pocket. It will then close off that pocket, essentially having swallowed those particles inside the cell, basically just by wrapping them up in cell membrane and pinching it off inside. Then the cell can do a number of things with it, depending on what it is. And this is a method that white blood cells will use when they're eating things that aren't supposed to be there, like bacteria or viruses or things like that. So in that case, they would take those particles into the white blood cell and then add some very powerful digestive enzymes to it to kill it. Or maybe the cell in this case is eating something big. Maybe it's drinking. Maybe it's whatever it's doing. It's taking things into the cell in large quantities or that are large in size. Whenever the cell is done doing that, it's done whatever it needs to do, there might be some trash left over. In the case of the white blood cell, maybe it wants to get rid of those things that it broke down and destroyed. Or maybe the product of the cell needs to leave that cell and go somewhere else. So maybe you made something that needs to travel somewhere else in the body. In that particular case, we'll see something else happen that's quite interesting. So here are the different kinds of endocytosis, or moving things in. I mentioned cell eating, that would be phagocytosis. Cell drinking would be penocytosis. And then receptor-mediated endocytosis is some sort of trigger, some sort of chemical stimulus that tells the cell to swallow something particularly. But either way, it's moving things inside by forming a membrane wrapping around it and swallowing. In the opposite process of getting rid of something, again, whether it was a waste product or whether it was something that needed to go somewhere else in the body to be used, we would use a process called exocytosis. Exocytosis is basically a cell spitting something out. So the process is, in step 1, take whatever it is you want to get rid of and wrap it up in membrane. And you'll notice the membrane there is the same structures that you find on the cell membrane. And we've already said when fat things come in contact with each other, they dissolve into each other. So that means the membrane that we made around this batch of stuff will fuse or join up very easily with the outer cell membrane. So in step 2, you see it fusing. In step 3, you see that package actually opening up. In step 4, the contents being released. In the bottom picture, there's a real cell doing this. So basically, that's package it, move it to the outer surface, and spit it out. So you can move large quantities of something, or you can move large sizes of something this way. So exocytosis is basically the opposite of endocytosis. And cells use perhaps both of these on a very regular basis. I don't want to spend too much time worrying about this particular graphic here, other than to introduce the idea of a membrane potential. When we talk about muscle contractions later on, when we talk about nerve signaling later on, this concept becomes very important. Perhaps you've heard of sodium and potassium pumps. Basically what they do is they allow sodium and potassium to move back and forth from inside the cell to outside the cell. And when they do that, that causes the generation of an electrical current. So in this image here, we have positive electrical charge on the outside, negative electrical charge on the inside. If we move a bunch of our potassiums here, the K pluses, if we move those inside the cell, that will then cause that electrical current to go from positive outside, negative inside, to flip, to become positive inside, negative outside. And when those charges flip, that generates electricity. And electricity is then capable of traveling along the cell membrane and doing something later on. So when we get to muscles and nerves, that generation of electrical current actually stimulates a signal to move, causing either muscle contraction or a nerve to send a signal. So the general idea here is these proteins here generate electrical current, and later on we'll dig more deeply into what that current is good for. Let's talk about DNA. You know DNA is important, but perhaps you don't really know how it works. You're not alone in that. No one in the world fully understands DNA, but here's the basics. DNA, as you can see up here at the top at number 1, is a double helix. So it is a double strand of material that is coiled up in that helical shape. Often in the cell, the DNA is found in the form of a chromosome. And you've heard of chromosomes before. And basically those are just tightly bundled segments of DNA. So when cells divide, they need to bundle that DNA up very nicely. So let's think for a minute about DNA and how it works. When we look at the cell cycle, this is the steps that a cell goes through in its lifetime. So we start with G1. And G stands for growth. So in growth 1, cells are doing everyday cell things. They're metabolizing, they're staying alive, they're repairing themselves. But they're also undergoing some growth in the form of making more organelles. In your reading assignment, you'll read all about the organelles. The mitochondria, the Golgi apparatus, the rough and smooth endoplasmic reticulum, the nucleus, and all that good stuff. So it's making more of most of those things, so that if it does divide, there's enough stuff to go around. So each of the new cells you might make at the end of this process have enough mitochondria, have enough Golgi apparatus to do what they need to do. So all of that happens in G1. Then in the next step, S phase, the S stands for synthesis. And this is where normal cell function continues, but the DNA is doubled, so that each resulting cell would have a full set of DNA. How many of you know how to synthesize or make DNA? No virtual hands going up here, I'm sure. That's okay. It's very simple to learn the basics. Do keep in mind that in reality, it is a very, very complicated process. Let's take a look at DNA. DNA has 4 ingredients. It has A's, T's, C's, and G's. Adenine, thymine, cytosine, and guanine. And what we need to remember is that DNA is a double helix. We need to remember that A's match up with T's and C's match up with G's. So if in one of those strands we had an A, in the other strand, in the same position, we would have a T. If in one strand we had a C in a particular position, in that same position on the opposite strand, we would have a G. So let's make a theoretical DNA strand here. Let's say that's our one of our strands of DNA. Remember, DNA is a double-stranded molecule, so if our first strand reads what it does, what would the second strand read? Remember our pairing rules. A's match T's, C's match G's. So if we have an A in strand 1, we would have a T in strand 2. If we had a T in strand 1, we would have an A in strand 2. C would go with G, G would go with C, and now you're probably starting to catch on here to this idea. So now we have a double-stranded molecule. A's match up with T's in the opposite strand, C's match up with G's in the opposite strand. So we say that these double-stranded molecules, these strands, they complement each other. So here's our DNA molecule, double-stranded. In reality, it would be much longer than this, but this is the space we have to work with. In S phase, what we want to do is take these DNA molecules and double them so that each new cell will have a full set of each. You want to make sure that your DNA count is correct, otherwise the cell won't function well. So what's going to happen is there will be a special enzyme come in and unzip this double-stranded molecule, making it into 2 separate single-stranded molecules. And this process of DNA synthesis is called semi-conservative replication. The semi-conservative part means that half of the old molecule is used in the new molecule. So we'll take the old top strand and we'll build a new matching bottom strand, which would then read, this should look familiar, it should read exactly the same as our old bottom strand did. And that's the whole point. You want to make new strands of DNA that are exactly the same as the old strand was. So here is our new DNA molecule. We've got half of an old strand, half of a new strand together, and so that's our new strand there. And so now we need to go down here and build a new top to match the old bottom. So it should read exactly the same as our old strand did. So now we've got a new molecule at the top that's half old and half new, and a new molecule at the bottom that's half old and half new. Now some of you might be raising your virtual hands right now and saying, something's not right here. I think you maybe made a mistake. But remember, I never make a mistake because I know everything. It's a nice thought, but not obviously realistic. But perhaps what you have noticed is that down here at the new top strand, instead of reading CAG like the old strand did, it reads CAC. That's a mistake. That's called a mutation. So basically a mutation is when the DNA is misspelled, the right ingredient did not get put in the right place, so you can force things into the wrong position. The good news is there are a number of proofreading mechanisms in the DNA structures that say that's not right, that's supposed to be a G, and they can go back and fix it. So most of the time mistakes that are made are found before it's too late and corrected. Later on we'll get into some ideas of where mutations might be a really, really bad thing, or maybe make no difference at all if they aren't caught and fixed. But for now, let's say mutations do not do good things for you. So this is S phase. So we started out with the double-stranded black-colored molecule. We ended up with two strands exactly the same as the old were, half old, half new, and that's S phase. Now that sounded very simple, and it is. Do keep in mind that in real life there are a lot of other little intermediate steps going on, loads of different enzymes involved in the process to get it done. So to appreciate now, it's complicated, but it's actually fairly straightforward as well. So back to our cell cycle. Let's say we've doubled all of our DNA and life is good there. In G2, we still have normal staying alive functionality going on there, and the final steps in preparation for cell division. So the whole point of a cell is to do its job, but also hopefully to make another cell, or perhaps two other cells, before it dies to replace itself. So if you're wanting to get bigger, you actually want one cell to turn into two, two to turn into four. If you're hoping to just maintain, you want to, for every two cells you lose, replace them with two more. So whether it's a growth or a maintenance process, it needs to occur. Once G2 is finished, then we actually go into the actual process of division called mitosis. So G1, S, and G2 are collectively known as interphase. For an average generic cell, about 90% of its life might be spent in interphase, not actually dividing, but getting ready to. Roughly only 10% of that cell's life would be spent in the mitotic phase, in the actual processes of division. Obviously, this will differ based on what kind of cell you're talking about. If you're talking about a muscle cell, the need for muscle cells to divide is relatively low, so you would see a relatively slow process. If you were talking about a skin cells actually need to divide very rapidly because about once a month you replace your entire outer layer of skin. So that would need to be a constant ongoing process to be able to keep up with the loss of skin. So different tissues would have different rates of division. In this division process, there are 4 steps: prophase, metaphase, anaphase, and telophase. I personally lump cytokinesis in with telophase and call it all telophase because I don't see a need to make it any more complicated than it already is, but just keep in mind cytokinesis, part of telophase. Now what's not shown in this particular visual is the idea of G0. We had G1, we had G2, but there is the idea of a G0, and what happens with a G0 is at some point during growth 1, there is a arrow that would branch off to the side where the cell exits interphase. In that, it doesn't prepare for division, it just lives. Brain and spinal cord cells are prime examples of cells that enter G0. So once your brain and spinal cord was fully grown and developed, all of those nerve cells went into G0. So they're alive and they're doing normal stay alive things, but they are not preparing to divide, and they will never likely divide again. This is why spinal cord injuries and brain injuries are such a big deal, is because those tissues do not have the ability to replace and repair themselves. So damage that occurs there, loss of cells that occurs there, is a permanent situation. If we could figure out how to take G0 cells and turn them on so they go back into active interphase again and then are capable of entering the mitotic phase, then we would be able to repair a lot of those problems. But we simply don't know how to do that. So for right now, G0 cells, if damaged, destroyed, or killed, it's permanent. So here's that DNA replication process going on, taking an old strand, as we see in the bottom there, and matching up new strand with it on top to get what you want. So that's a little bit prettier version of what we just did with our A's, T's, C's, and G's a minute ago. What is DNA? You know it's the genetic material of a cell, but what is the point of the genetic material of the cell? And the simple answer is, it is the instructions for how to make proteins. And really nothing more than that, just instructions. And those instructions are stored in the nucleus of the cell. So let's compare this process we're about to engage in here to the idea of making a Corvette, which occurs at the Corvette assembly plant just down the road here in Bowling Green, Kentucky. So with this idea of DNA, it is the master plans, the master blueprint for how to make a protein. So if we're thinking about the Corvette, we're talking about the master design plans for a Corvette. Would you want to use the master plans at the actual site of manufacture? Would you take the original drawings and designs for a Corvette out onto the assembly line and lay it down next to a welding robot, or a painting robot, or any kind of manufacturing process? The simple answer is, of course you wouldn't. And that's because if that master copy gets damaged or destroyed, then you're completely out of luck. Your cells approach things the same way. The DNA is going to be stored in a safe place, just like the Corvette plans are. And I can assure you the original Corvette plans are probably nowhere in the state of Kentucky at all. And perhaps very few people would actually be able to tell you where they're at. So in a cell, if we keep the DNA safely in the nucleus, then it's not likely to get damaged. Keep in mind we'll actually build the proteins from it out in the cell itself, not in the nucleus. But we have to somehow convey that information from the nucleus to the manufacturing facility. So for the Corvettes, there are copies of those plans. Those can actually be taken out onto the manufacturing floor, and if they get damaged, so what? It was a copy. We can make more copies. In the world of DNA, we're going to do the same thing. We're going to make a copy. The copy is then disposable, and the copy can be used to build the protein. So the first part of building a protein happens in the nucleus, and it's called transcription. Transcription is basically taking the DNA instructions and making a disposable copy called mRNA. M stands for messenger. So it's a messenger RNA. And in the world of RNA, it is a very, very similar structure to DNA with one big difference. In DNA, we had A's and T's. In RNA, we don't have T's, we have U's. So A would match up with a U instead, uracil, instead of thymine. C's and G's would still go together, but A's would pair with U's instead of T's in the RNA world. And we'll see that happen here in a little bit, and hopefully that will make sense. Once that disposable mRNA copy is made, it leaves the nucleus and enters the next step of protein production called translation. And transcription basically is transcribing from one dialect of a language, the genetic language dialect of DNA, to the genetic language dialect of RNA. So it's got minor modifications of the replacing T's with U's. Translation goes from one language to a completely different language. So that's going to go from genetic language of the mRNA to protein language of the finished protein. So there are much more significant changes that occur here. Let's actually take a look at that. Before we do that, though, let's look at those actual stages of cell division. We talked about interphase, and in interphase and in all the stages of cell division, you look at the nucleus. So in the nucleus here on the left, this actual real photograph, this blue ball here is the nucleus, and we really don't see much there. If we go over and look at the drawing, that's because the DNA is very loosely scattered throughout the nucleus and doesn't show up as visible strands of material. So that's the G1, S, and G2 phases would all look like this. In prophase, what happens is the DNA starts to bundle up into chromosomes. Because we're going to take this now doubled amount of DNA, and we want to have 2 new cells come from this, each having the proper amount of DNA. If we don't organize and separate the DNA properly, we'll end up with mistakes. And if you get too many chromosomes in one cell, not enough chromosomes in the other, that leads to disastrous results. Often in the form of the cell spontaneously dying because its chromosome count wasn't properly done. So in prophase, we take that very loose, scattered DNA that we saw in the interphase nucleus and bundle it up into chromosomes. The idea is we have fewer bundles of chromosomes to work with. It's easier to sort them and keep them separate. So what you see here in the real cell, you start to see these little squiggly lines. That's the formation of chromosomes. So all that DNA is bundling up into what under a microscope is now visible. The second thing that happens in prophase is the removal of the nuclear envelope. So that membrane that was around the nucleus has to go away. Otherwise, all that DNA is stuck in the nucleus and we can't distribute it to the new cells we're trying to make. Here in what they label as late prophase, which simply means we're just about done with it. Very nice visible chromosomes there. We don't see a nuclear membrane anymore because that's been dissolved. Now we're ready to go to the next step, and here we're going to take all of those chromosomes and line them up along the center of the cell. Think about what your favorite time of day was in grade school, and most of you are probably going to say recess. When you got to go out on the playground and run and play and do kid things. And when it was time to go back into the classroom, what did the teachers do before you went back in the room? Probably they said, line up in your rows. So each grade would line up in its row, and you'd have a row of kindergarten, a row of first grade, a row of second grade. And the point was so that when you go back into the classroom, the right students go to the right places. You didn't want to have kindergartners in the first grade room, first graders in the kindergarten room, and second graders scattered all 3 places. That would be completely messy and counterproductive. So forming the lines helps to make sure that if one line goes one place, you probably got the right people in the right place. In metaphase, by lining up on this line, the top half goes to one end of the cell, The bottom half goes to the other, and if you separate properly, the exact distribution of chromosomes goes to the right place. If that happens properly, then each new cell that you're making has a full set of everything that it needs to have. If you make mistakes here at this point, that leads to, again, disastrous things of the cell dying or certainly having some significantly impaired function because the chromosome count wasn't right. Typically, a chromosome count mistake ends in spontaneous death of a cell. So clearly a big deal. The next step in mitosis is anaphase. That's the actual pulling of half of the chromosomes to one direction and half of the chromosomes to the other. And in my view, this is the most interesting looking stage of mitosis because it looks like two hands pulling away from each other. In this case, each hand has about 30 fingers, or it appears to have about 30 fingers. It would really have quite a bit more than that, but the point is it looks like fingers trailing along as the hands start to pull apart from each other. You're starting to pull one set of chromosomes to one end, the other set to the other end. In telophase, you take those hands of DNA and you undo prophase. So remember, in prophase, we formed chromosomes and we removed the nuclear envelope. Now these chromosomes are going to start to uncoil, so they're available because the cell cannot read and use a chromosome when it's all bundled up like that. So we're going to undo the chromosome bundles, and we're also going to reform the nuclear envelope so that we have two separate nuclei. The last piece of this puzzle is often labeled as the cytokinesis, and basically that is the growth of a membrane down the middle of this now elongated cell, physically dividing it into two separate cells. Once that membrane completely separates the two cells from each other, they are now two new cells. And they're much smaller than a full-grown cell would be because they were just formed, but they have all the pieces they need. They'll continue growing to reach full adult size for that type of cell. So prophase, metaphase, anaphase, telophase, and that gets us two new cells. So back to this idea of making a protein. If we start with the DNA at the top here, we're going to uncoil the DNA because we can only use one strand at a time. So we're just going to read one strand here. And so we see our string of T's and A's and C's and G's. And we're going to form the mRNA copy in transcription. So we're going to match up so we can see here in our DNA we had a T, which we know matches with an A. So here's the A in our mRNA. Our next letter was an A, and that normally would match with a T, but remember in RNA we have U's instead, so there's the U. We have a C, would match with a G. And we'll go down the entire strand of DNA, copying, or matching up rather, C's with G's, and A's with U's, or T's with A's. So we're making the mRNA the disposable copy. So our TACGGT became, or matched up with, AUGCCA, and on down the line. That mRNA copy then leaves the nucleus, goes out into the cytoplasm, or the juices of the cell, and that's the instructions. So the molecule that comes along and reads the mRNA instructions is called a tRNA. And that T stands for transfer. The job of a transfer RNA is to carry an amino acid, a protein ingredient, and put it into the protein at the appropriate location. So if we go back to our Corvette manufacturing example, when in that process do you want the battery? At what point in making a Corvette do you put the battery in? Well, the answer is it's the last piece of the Corvette that's put in place before it rolls off the assembly line, which means we don't want the pallets full of batteries anywhere in that production process except for at the very end of the assembly line. If we had pallets of batteries stacked next to the welding robot, that's in the way. That's useless. It's not doing any good. If it's in the middle of the spray painting process, that's not useful. If it's in the middle of installation of the engine and transmission, that's not useful. So we don't even want the batteries in the building until the last piece of the puzzle is ready. So the job of the tRNA is to carry the right piece and insert that piece at the appropriate time. So we have our instructions of mRNA, and if we have AUG starting out the strand, we want to find what tRNA will match with AUG. So AUG would match UAC. Remember, A's and U's match, C's and G's match. So AUG matches with UAC, so that particular tRNA will carry the appropriate ingredient for that location. The question is, what's the appropriate ingredient? And that is something that you need a chart for, because I don't know of anyone who's ever memorized all these. I suppose you could if you're really bored, but surely you have something better to do. In the 11th edition, it's on page 103 in your text. In the 10th edition, it's going to be back in the back of the book with all the appendix materials in an A-something page. But it will be a chart that has a big list of tRNAs in it. And it will tell you which amino acid each tRNA carries. So if you go find that and look up UAC, or sorry, rather look up AUG. So look up AUG in the chart, you find that matches with, or carries methionine. So the tRNA that matches AUG will carry the ingredient methionine. The tRNA that matches CCA in the second piece of mRNA there. So if we would look up CCA, we would find that it would carry proline. If we look up UCG, that would tell us that we're looking for serine. And so that tells you which amino acid goes where. And so by the end of this process, hopefully we didn't make mistakes. But remember the idea of genetic mutations. If we misspell the DNA, that's misspelled our protein instructions. And that will mean that when we're matching up tRNAs, that we don't get the right final ingredient. And what we would then have is the wrong ingredient in the wrong place. And something as simple as one letter being off could result in one ingredient being off of a protein that perhaps has hundreds of thousands of ingredients. But even if one single ingredient gets off, that could cause serious problems with that functionality of that end protein. So mistakes in the DNA very at the beginning of the process can lead to mistakes in protein later on. And that's why it's such a big deal, is because proteins don't get built correctly, and then you don't get the functionality they were supposed to have. You will always start proteins with methionine. So you'll always find the starting section of DNA and mRNA will always call for methionine. Once you get started, you go until you reach something. And the example here is UGA. And if you look up UGA in that chart, it says stop, which means stop. So you start when you have a large string of methionines in a row, and you stop when you have one stop message. If you're looking at that chart, what you'll see is that proline has 4 different ways that it can be spelled. You can have CCU, CCC, CCA, or CCG, all giving you the outcome of proline. What this really means is that as long as you spelled it CC something, you'll get the right outcome. It didn't matter whether it was an A, a C, a U, or a G, you'll get the right outcome if the first 2 were right. Looking at that chart, there are really only 2 amino acid ingredients that only have one spelling. That would be UGG for tryptophan and AUG for methionine. So everything else has at least 2 different ways you can spell it and still get the right answer. If we look at arginine, there are actually 4 different ways arginine can be spelled. CG anything, and half of the AG anythings will also give you arginine. So that suggests that arginine is a very important ingredient. And the more important the ingredient is, the more different ways there will be to spell it. This really means even if you had some typos and mistakes along the way, as long as the mistake happened in the 3rd letter of each word, probably doesn't change the outcome. So redundancy prevents problems. The more redundancies there are built in, the fewer problems will actually be exhibited. Which means to start a protein, there's only one way to spell it. To stop a protein production process, there are 3 different ways to spell it. UAA, UAG, or UGA, all 3 of which will give you the stop word. So that means it's more important to be able to stop a protein's production at the right place than it is to be able to start. So it's better to not start when you shouldn't, or even if you should, than it is to not stop when you should. Let me make that a little bit more clear. If you fail to stop when you're supposed to, that's called cancer. Cancer is protein production that has not recognized the stop word, and it's building when it doesn't need to be building. So if you were supposed to start and you didn't, then you're short on that protein. If you're supposed to stop and you didn't, that quickly becomes labeled as cancer. I think that sounds a lot more scary to you than a simple protein deficiency. Hopefully these concepts and processes make sense. Your textbook has a number of very beautiful drawings that go through this protein production process. If you have questions, as always, post those in the discussion board or ask them in our live session, which will be coming up on Thursday. Make sure you check Blackboard for specific instructions on that, because our live discussion platform is probably going to change from Blackboard Collaborate to something else that hopefully will be a little bit more user-friendly. Again, if you have any questions, be sure to ask. Have a wonderful day.